Hi everyone, my name is Anuj Dev Singh. I recently graduated from IIT Madras on BS degree at Data Science and Applications. This is my research path in Build with Cloud Science Hackathon. All AI models, the problem is all AI models like AlphaGenome and Borzoi can read DNA and predict what's happening in the genome daily well. Everyone excited about that, but I found they all fall at the one question that actually matters for what this disease. Let me show you what fails and why. The most disease are basically the most disease are causing the DNA changes aren't inside the genes. They are in the tiny switches that control DNA genes when a patient carries one. A doctor needs one thing. Does it turn the genes up or down? That direction decides if it's harmful. So the real question is, can these AI models predict the directions of that direction that what I tested? So basically, this is a benchmark that I'll show you. Yeah. So I built a fair test. I collected the real variants where the directions was already measured in the lab and the human microglia in the exact brain immune cells. These models are trained on. I split the data by the whole chromosome, so a model cannot cheat by the memorizing nearby variants. Then I scored every model on the same hidden sets. Okay, now I'll show you the results what I got. And the results I got as this one. These are the results. The alpha genome predicts really well 50%, 55%, Borzoi is 56%, and the same distribution. So basically, and the random guessing is 56%. So they are basically flipping a coin. And the key part is I even trained a model directly on the data, and it's also landed at a chance. So this isn't a big model. We'll fix it. Will a big model will fix it a problem. The direction just isn't in the DNA sequence. So what do they fail? Why do they fail? That is the main question. And so in the same, so here's the research I found and research I did, and I found the answers for the why did they fail. So in the same 95 peoples I asked, when the variant opens the DNA, basically open up the DNA, yeah, opens like DNA, does the gene go or rep? The answer is no relations. The number is 0.506, a perfect coin flip. And think of it at a door room. Chromatically accessibility is whether the door is open. Expression is whether the people actually walk through. I found that knowing the door is open tells you nothing about whether anyone walks nothing through, and they are independent. So a model that predict only one of them can never gives you the direction of the other, and that's the wall. So yeah, so basically, so only one I tested on the one real variant, and that's a famous one, Alzheimer's variant, that is RS 6733839. And atop Alzheimer's risk variant near the gene BIN1. It's opened the DNA, it's raised BIN1 expression, but it represents an isolated switch in the lab test. 3 layers, 3 different answers. So every model looks right on one and wrong on another. Then using the AI structure predictions, bolts 2, I showed the risk version grips the protein MEF2A tighter, the 12% more contacts. It's creating a new binding sites. So yeah, so the takeaway is don't trust a single layer AI model directions, goals on the variant that matter most for the diseases. And it's all here. The benchmark, every score, the paper, fully open and reproducible. My contribution isn't being the model, it's showing you why they fail and giving you the field a clean way to measure it. Thanks for watching.